The next one we're going to go through is our ATPases. The prototype that we're going to use for this is a V-type calcium or V-type sodium potassium ATPase, or a pump. Now, why are these pumps so important? Well, this is important because the last two things we went through, pores and ion channels, needed to have a concentration gradient for there to be any transport across the membrane. Here, we are creating the gradient, or we are pumping against a gradient. So we don't need to rely on a gradient. We can do it ourselves by using energy to get us across that particular cell membrane. So that's why this is called active transport. And this is used to either establish a gradient or move something against a gradient. Okay, besides having ATPases in places like the plasma membrane, we can also use ATPases in other places, such as the endoplasmic reticulum. There's a certain kind of calcium pump located on the endoplasmic reticulum of muscle cells that allows us to pump in calcium. And why this is so important is this is going to help us relax a muscle by removing calcium from the cytosol of that muscle cell. Now, what are the important aspects of having these particular ATPases? Well, the first thing to think about is there needs to be ATP hydrolysis occur. Now, this occurs both on our pumps as well as on our ABC transporters. And these are not pumps in themselves, but still require ATP hydrolysis or energy to cause the transport process to work. But let's get back to our prototype. Usually for our prototype, what we're trying to do is pump a certain number of ions across the cell. For this sodium potassium ATPase, we're going to be pumping an unequal number across the membrane. So 3 get kicked out of the cell and 2 potassium move into the cell. This unequal number, though, creates an electrogenic response, which means that there's going to be an electrical gradient formed because you're losing 3 positives and gaining 2 positives. That means there's a positive difference there. As that difference occurs, you're going to create an electrical gradient. So let's go through that prototype in more detail. There's a number of cyclical processes involved here, and we're going to take these one by one to explain to you how this process works. The first thing we're going to talk about is to think about the number of different ions that are exchanged. So this again is a V-type pump, extrudes or pushes out 3 sodium and brings in 2 potassium. The other things to think about is where this is primarily located. Usually this is going to be on the basolateral side of most epithelial cells. And we'll keep talking about epithelial cells throughout this particular course. But these cells are usually located along a junction in which you're moving something from one side of that cell across one membrane into the cytosol, then across that membrane on the other side to be moved into somewhere like the blood. It involves a number of steps, in which here we have 8, and we'll try to animate these for you so you really understand how this sodium potassium ATPase works. Okay, now that we have the basics of the sodium potassium ATPase down, let's now take each one of those steps in turn. So let's first start off with the basic portion of the pump. So we have ATP bound to it, we have the outer gate closed, and we have nothing in the pump yet. We now have sodium that enters into the pump, in fact 3 sodium to be precise. The next thing that happens is the hydrolysis of ATP. Hydrolysis means we break down ATP into an ADP and an inorganic phosphate. The inorganic phosphate stays bound. What this also does is close the inner gate. Now the outer gate opens through a conformational change, and that allows sodium to leave. This then allows the potassium, in fact 2 of them to be precise, entering into the pump. Then inorganic phosphate leaves the pump, closing the outer door. And now ATP binds back to the pump, which opens up that inner gate, allowing potassium to go into the cell. And that sets up the sodium-potassium ATPase pump cycle, where you need to have ATP bound, broken down, sodium entering, sodium to exit the cell, potassium to enter the pump, and then finally potassium to enter the cell. And it's very important to have that process working correctly to open and close the various gates at the appropriate time. It's a great example of a sodium-potassium ATPase V-type pump.